Welcome student. In the next lecture of our topic which we are dealing about is the secondary growth. So in the last class we have talked that how secondary growth formation takes place in stem and root, and when we talk about secondary growth in stem, the major portion of that is occupied by the secondary xylem, which is known as wood. So when you would have seen a plant also, a tree also, which has a very large diameter, and you would have seen that the plant would have is cut and you observe the stem diameter. This is all what you see is basically the secondary xylem, and this is the most economical product. It is most commercial product which is obtained from a plant, and it is used for making various things like furnitures. Everyone will have things made up of wood in their houses. So let's try to discuss in somewhat detail about the nature of this wood and how the composition of that wood varies, and how different types of wood is present in nature, and what different classification has been given to them. Now see, wood is the product of secondary growth. Okay, it is formed only after secondary growth. You will not find wood during primary growth. Okay, and it is the multi-year accumulation of xylem. See, wood is basically secondary xylem, and it's not that just once the cambium is active and secondary xylem formation is there and you find a wood. Okay, it's through years. Okay, multiple years, several years they will take, the plant will take to accumulate that big structure which is having a big diameter, okay, and which is called as wood, okay, that hard structure. So the purpose of this wood is to provide support to the plant and to transport water from the soil to the leaf, because definitely what role they are playing in the plant is to transport. It is the thing which we are taking from them and we are using, that is the extra benefit that the plant is giving to us. But for them, the wood is there for mechanical support, it is for better transport and conduction. It is the most widely used plant product. Okay, so the plants, from we obtain large number of things. Okay, we obtain food, we obtain medicines, many other things also, but wood is also one of the largest material which is obtained from plant and which is used for our human purpose, which humans use for their own purpose. Okay, they use them in making furnitures and various other things. In fuel also, we burn wood for fuel, even in electricity generation. Some some food supplements are also there. So in food also some sort of woods are also used. Now you would have heard of some names, like in India we have like teak woods are there, mahogany woods are there, you have heard name of sagwan, sesame. Okay, so these are all different types of wood, and then they have different properties, and because of that property they are used for different purposes. Okay, now the first important thing which we observe in a wood is presence of annual rings or growth rings. I think so that all you would have all seen those rings in a wood or in a tree. When you cut a tree, when a tree is cut, and then when you observe the cross section of that tree, you can see lot of ring like structures in a wood. Even in your furnitures also, when they are made up of wood, you would have seen some sort of lines on that. Okay, so they are all annual rings or growth ring. Now how these are basically concentric layers, like one after the other these layers are formed, and these are annual. Okay, means in 1 year one ring is formed, in the next year another ring will be formed. Okay, so one ring represent one seasonal increment. That means the area of xylem which is added between two rings is an increase which is occurring in one complete year. Okay, now the cambium is active, and how much secondary xylem is produced by cambium within 1 year is represented by one ring. Okay, the next ring will represent the next year. Now when you will see those rings, you will see two, even in one ring also. Okay, one ring also you will see two layers actually. One will be slightly darker, okay, and the other will be slightly lighter, and the lighter layer will be more in diameter as compared to the lighter one. Okay, you can see the darker region and the lighter region and the darker region. Okay, so this lighter region is known as spring wood or early wood. Okay, and it is more in diameter because see, in the spring season the environment is more favorable, more water is there, temperature is also very suitable, the activity of the cambia is very high, because definitely in all the favorable conditions you would have seen that the plant is growing very fast, leaves are coming very fast. So if leaves are coming very fast, they need more conduction, more translocation. For that, more tissues has to be, more secondary tissues has to be formed. So more number of secondary tissue or more xylem, secondary xylem is formed. So that part of the wood is known as spring wood. That means in this, the lighter one is known as spring wood, and the other one, the darker one, is known as autumn wood, because in autumn or in winter conditions are not so favorable, and because of that cambium activity is not very high. So the activity is very low, that is why you have a very narrow zone, and then this is also known as late wood. Okay. Now in spring wood, this figure you can be very sure, you can see very clearly that this is the spring wood and this is the autumn wood. So these both woods actually correspond to one season. Okay. So when because in spring season and the activity is more and it is accumulating more xylem tissue, in autumn more the xylem tissues are less accumulated and they are more compact, they are more lignified, they are more narrower. Okay. The elements like trachees and vessels they are more narrower, but they are more thicker. Okay. That is why you see a very darker structure. This layer is dark as compared to the spring wood. Okay. Even trachees and fibers, more lignified tissues are more abundant in autumn wood. Okay. That is why the layer appears darker, whereas spring wood, the layer is wider and the tissues, the tracheary elements, that the xylem and trachees are more wider. Okay. They are less thicker as compared to the autumn wood. So that is why you see a lighter structure. Now this corresponds to the age of the plant, the annual ring, because in each year one complete ring corresponding to the autumn and spring wood, that complete ring formation takes place, and therefore this corresponds to the increment in 1 year. So in that way, if you count the number of rings, you can say, you can even calculate the age of the plant, at least from the time from where they have started the secondary growth. And generally in woody plants or woody dicots, the secondary growth starts very early. As soon as some of the primary tissues and the plant have attained certain height, then they start forming secondary growth. Now dendrochronology is completely a branch of science deal with this counting and analyzing this annual growth rings of trees. So in that way you can find the age of the tree. So it also tells you something about the nature of the climatic condition of that period of time. See, many trees are 100 years old or more than that. So you can even somewhat get some idea about the climatic condition of that time. Now the conifers and many temperate dicot have prominent ring. See, you can imagine that, see, if there is a temperature or if there is a climatic difference, then only you will get these different variation in growth ring. But if the temperature or the climatic condition is constant throughout, then you will not get an appearance of these ring-like pattern, because this is only the difference in the cambium activity. But if the climate is uniform throughout the season, the cambium activity will also be throughout. So in some tropical systems, you will not find growth ring. Like this is a tree which is not actually showing any growth ring, because it is grown in those conditions where climate is almost uniform. So in that case, you will have some smooth grained like appearance. Okay. Now these figures also tell you that what is the difference between the spring wood and the summer wood or the autumn wood. Here also you can see a large patch of early wood or spring wood and a small patch of late wood or this autumn wood. Okay. Here also you can see the vascular cambium is, see, such a large patch is occupied by the secondary xylem. Okay. And here you can see small amount of phloem is here, but these sizes can vary depending on the plant species, and then later core cambium will also become functional, some sort of bark or periderm will also be formed to increase the diameter of the outer layers. Now another term you would have heard of is the softwood and the hardwood. Now in the commercial sense or in the common sense, softwood means gymnosperm wood, whereas hardwood means angiosperm wood. See, when you hear of softwood, that means it is obtained from gymnosperm, because gymnosperms also have a very huge diameter and many of the woods are also obtained from gymnosperm, okay, like spruce, pine, many species of gymnosperm, and hardwoods are angiosperm. Now they are known as hardwood because there is a difference, okay, we have studied before that gymnosperm doesn't have vessel, whereas no vessel, okay, and vessels are present. So that's make the difference. Because of this vessels, the hardwoods have very high density than softwood, and because of that they are known as hardwood, because they are more denser as compared to softwood. And the softwood also have typically slightly faster growth rate as compared to hardwood. Now fibers are also more in hardwood, so that all make it more dense, means more mass is there in those hardwoods. Softwood example like pines are there, then spruce, Douglas fir, junipers, okay. Similarly hardwoods, all the dicot plants are there, maple, oak, teak, all those things are there, okay. Due to the presence of vessels, the hardwood is also known as porous wood. See, basically vessels are what? That multiple times I have told you, these are pipe-like structures, so definitely they are empty pipes. So when you will cut it, they will appear like holes. So you can say that these are pores, very big pores are there. So these hardwoods are also known as porous wood. There could be a ring porous structure when like in Acacia fistula and Dalbergia siso, see that we will discuss later also, that in which these vessels are confined to a particular region and they form a circle, they are known as ring porous, but when they are found diffuse in the entire region, when they are present in entire region, they are also known as diffuse porous, okay. Now another term is there, sapwood versus hardwood, okay. What is sapwood? See, sometimes when you would have seen the cross section of a tree, okay, when the tree is cut and you observe it, you see that in the center, the wood appears very dark in color, okay. The heart of the or the center of the stem, the wood is quite darker as compared to the outer very light region, okay. So this is actually age related, and it is because when the plant grows older, the inner region becomes more darker in color, and it is because it becomes lot of many compounds accumulate like terpenes, polyphenolic compounds. With time they accumulate in those tissues, they actually lose their capability to conduct water, they become they have less water, and mineral reserve, they do not participate in translocation, because now they contain lot of things, lot of many other compounds enter inside and actually they block the entire vascular system. So now they are not conducting. Type of wood is known as hardwood. Their color is because of accumulation of all these things. Sapwood is lighter in color, and it is because it is more active, it is more involved in conduction of food material and conduction of water. It has more level of moisture in that, because since it is conducting water, so it has more water. It is involved in active transport, and it is also designated as living wood. See, living doesn't mean that it doesn't have any dead tissues, but living in the sense that it is performing its work, okay. It is involved in conduction, and it does, and because definitely this wood would have been formed earlier, okay. So now with age more things have accumulated inside it and they have blocked it, so now it is not conducting anything, more materials are there. Outer one In the outer tissues, it is more conductive, that's why it is more lighter in color, because it does not have any other depositions of tannins which are colored, which are like dark in color, like polyphenolic compounds. So, they give all those peculiar color to that hardwood. So, in this way you can differentiate between sapwood versus hardwood. Now, sapwood, when we when we make furnitures, many a time hardwood wood is preferred because it contains less moisture. It is it is not as prone to decay as compared to sapwood, and sapwood, since it contains water, so there are chances that with time it might shrink, but hardwood will not shrink because the moisture content is very low in them. Now, conifers has tracheid, parenchyma and rays. So, vessels are actually absent. Vessels are absent. Okay, so this is very important thing. So, when you will see the cross section, okay, this is TS is the transverse section. Okay, so this also many a times I have told. So, there are different ways of cutting a tree to analyze the arrangement of all the vascular tissues in that. See, there are basically 3 ways of cutting a tree. So, see, one way is I have told you that it is the transverse section, you cut it and you just observe it from the top. So, this is the TS. Then one is the RLS, that is the radial longitudinal section, and this is the tangential section. Okay. So, how when you cut them into different sections, you better observe all these different arrangement of trachees, vessels and rays. So, when you observe gymnosperm, see here also in transverse section you cannot see any vessel like thing. Okay. And this is the autumn wood, this is the late wood and this is the early wood. You can clearly see the difference. You can see a big patch of early wood and a small patch of late wood, okay, which are thicker and they are more condensed, having more fibers, that is why they are appearing more darker in structure. Now, when you cut radial longitudinal section through radius, see this is all radius, this is all radius. So, if you cut anything in this plane and this plane, suppose if you are cutting in this and this plane, okay, a small section you are cutting in this. So, longitudinal section you are cutting, okay, radius, through radius, passing through the radius, you are cutting a longitudinal section. So, in this you will see arrangement of tracheids, you will see longitudinal arrangement of tracheids, bordered pits will be there, and then radial arrangement of parenchyma you will see, because this is oriented in the vertical direction. So, here also when you will cut radial longitudinal section, you can see tracheids are there, okay. All the tracheids do not have they are not connected or they are single celled with tapering ends. Then you have bordered pits in them. In gymnosperm, mostly the tracheids you have bordered pits. So, you can see here this is an enlarged view showing the bordered pit, and this is the radial medullary rays or ray parenchyma. Okay. So, this is running vertically, this is running radially. So, in this way in RLS you will see this type of pattern. Enlarged view also you can see tracheids here, you can see parenchyma in this direction. Okay, this is the parenchyma, this is the tracheids. Okay, tracheids vessels are not there in gymnosperm, so you will always see tracheids in them. You can also see some thick one structure known as crassulae or bars of Sanyo, which are cellulose thickening between pits. See, these are 2 pits and between them some cellulose thickening are there, which is known as crassulae. Similarly, one section is tangential longitudinal section. Now, you cut on the tangent, on this boundary, on the periphery, from the surface side of this cylinder, you cut a layer. You cut a layer from the surface, okay, not from the radius, okay. You cut a longitudinal section from the surface. See, in this way we can observe the medullary rays because they are radial, okay. So, they will end here. So, you can see the orientation, how they are oriented. So, in this section you can see the arrangement of the medullary rays, you can even see the bordered pits, you can see the torus in that. If you observe it like this, you will see, okay, you will see all those torus, and you will see medullary rays. Okay, they can be uniseriate, they can be multiseriate. So all this arrangement you can see in tangential section. Now, dicot stem has vessel. Now, vessels are very important, and there are various nomenclature to identify the structure of wood based on these vessels. This vessels could be called as ring porous. See, the presence of vessels will make them porous. Okay, they are known as porous wood because they contain vessels, pore-like structures. Okay, vessels are pore-like structures. Now, they are known as ring porous when these pores develop. See, during early season in the spring wood, the vessels are bigger because more favorable condition are there. More vessels are formed in the early wood. Okay, you can see here, and less vessels are formed in the late wood. These vessels now they are accumulated at one particular location and they form a ring-like structure. So, they are known as ring porous. In diffuse porous, in some wood it is observed that the porous is uniformly, see in the late wood also, in the spring wood also, you can see the porous vessels are uniformly distributed in the entire wood. Okay, similarly, you can see semi-ring porous when the pores are less grouped, like in this as compared to this one, pores are less grouped in the semi-ring porous. Similarly, in dendritic pattern, you can see that the vessels are branching out. Okay, the vessels are branching out from the early wood to the late wood. Okay, so this is known as dendritic pattern. However, intermediate patterns could also be observed. Now, these vessels grouping, some nomenclature, they could be grouped in different ways, like they could be solitary. See, you can find single vessels. Okay, single vessels you can find distributed in the wood, or it could be cluster. Now, clusters could be very few, like 2 or 3 cell cluster, or multiple cluster of these vessels you can see. So, there are even very large clusters of these vessels could also be observed in wood. Now, there are another way of classifying parenchyma. See, as I have told you that parenchyma of xylem and phloem, they both are distributed axially as well as radially. So, let us talk about the axial distribution. See, parenchyma could be distributed in different ways in the axial system. So, in this system, so how they could be? See, one is apotracheal parenchyma. See, vessels are there. Okay. Now, these parenchyma, small parenchyma, suppose you take just a small piece of this longitudinal cross section and try to analyze here. Now, these parenchyma are not associated with vessels. So, they are known as apotracheal parenchyma, whereas in paratracheal parenchyma, you will see that these parenchymas are attached with vessel. Now, it could be diffuse parenchyma. If in this parenchyma, these cells appear as single cells, okay, distributed throughout the growth ring. So, it is known as diffuse. In banded parenchyma, you can see that they form a band-like structure. Okay. Now, in this banding pattern also, or you can have boundary parenchyma if these are found at the beginning or at the end of the growth ring. Okay. Growth. Suppose this is a growth ring. Okay. At the beginning of the growth ring, if this parenchyma is present, it is known as initial, or at the end of this growth ring, if it is present, then it is known as terminal. So, this is the entire growth ring. But in this you can see that they are not attached with the vessel, whereas in paratracheal parenchyma they are attached with the vessel. Now, scanty, when few parenchyma is attached with the vessel, it is known as scanty, and in vesicentric parenchyma they form a continuous sheath around vessel. Okay, so they form a continuous sheath. In alliform, what happens? They form a continuous sheath, but they extend to form wing-like structure. Okay, they are attached with the vessels, but they also extend to form wing-like structure. So, this is alliform. Okay. So in this way we can classify axial parenchyma. Then ray architecture, see that also we have discussed several times that rays are radial conducting elements. They could be classified as homocellular. Sometimes some axial they are made up of living cells, living parenchymatous cells. Sometimes some dead, dead tracheids here means that they are non-conducting. It's not that they were living cells and now they have become dead. It's that they are not performing their function as in axial conduction. So they could be homocellular when only single type of ray cells are there, and heterocellular we call them when a mixture. See, mixture means some sort of upright and some sort of radial, but in mostly radial cells are there, and in between they might have some upright cells also. So it is made up of a composition of both the types of rays, and some trachees, dead trachees means which they are not performing in conduction, they could also be there. Now this also we have discussed, these rays are basically the medullary rays. Okay, so they could be distributed in different ways. So that also we have discussed, that either you can have very short rays of 2 to 10 cells, or okay, or they could be several rays like hundreds of cells could be aligned together, okay, to form a ray. And similarly you can have uniseriate, okay, uniseriate just single file, biseriate 2, and then multiseriate several files. So that all we have discussed before. Now another important concept in the last I will just try to discuss is about reaction wood. See, reaction wood is a type of wood which is formed by some reaction. Okay. So now wood in nature, it is subjected to various forces, like wood could be in some places because of some gravity, they could be bent, or some mechanical pressure is there. So because of that they slightly bend, or they are slightly oriented in some other direction which is not normal for them. So now because of that structural changes, because of those changes the wood undergoes some structural changes in its composition, and that change is known as reaction wood. Means the wood which is formed after those structural changes are known as reaction wood. Now this reaction wood is present in gymnosperm also, in angiosperm also. However, they originate in slightly different pattern. Indole acetic acid is it's an auxin, okay, which is very important in formation of this reaction wood. Now in conifers like in gymnosperm, the reaction wood is known as compression wood, and in dicot it is known as tension wood. See, suppose this is a wood, okay, this is a tree, and if we try to pull it, so what will happen? It will try to bend in this direction because of that pull. So it will now be in a tension. On this side in compression, this side it will be in tension. Okay, so compression and tensions are both things which happens when we apply force. So this portion, when wood develops on this side, it is known as compression wood, and wood when it develops on this side, it is known as tension wood. In gymnosperm, compression wood, like in gymnosperm, see this wood, what happens, you can see here that now the growth ring formation is not exactly uniform, it is more on one side. So, and in dicot also you can see that it is more on one side. So in dicot it is generally on the upper side, so it is known as tension wood on this side. So it helps in pulling the stem back towards its normal position. And in gymnosperm also it is on the other side, it is on the lower side, which might be useful in reducing the pressure which the plant is getting exerted. So in this tension wood, when we talk about this wood, we have discussed about gelatinous fibers. Now these type of fibers appear in tension wood. These are also some sclerenchymatous fibers only, but in this case they have greater amount of cellulose, okay, and a reduced amount of lignin. So these type of wood accumulates in response to some forces, and they are known as reaction wood. Just a small thing, we will discuss about tyloses. What are tyloses? See, in some vessels, what happens? when some infection or occurs, arises, or when some mechanical damage is there, when some drought condition is there, these vessels the cells try to block these vessels. Okay, so like suppose some infection is there, and if the passage is blocked, the pathogen will not able to enter it, and it could not pass through inside the cell through that passage. Or if some drought condition is there, the vessels will be blocked so that no further water conduction can take place. Similarly, in case of mechanical damage also, it occurs to vessel or something happens there, so it should not proceed to other cells. Some sort of structures originate which actually blocks the vessel. Now, they are known as tyloses, and they are protoplasmic extensions of adjacent xylem parenchyma. So, you can see here that this is the parenchyma, okay. This is the parenchyma, and this protoplasm or this parenchyma enters and it forms a balloon like structure. So, this structure entirely blocks the vessel, okay. So, it entirely blocks the vessel so that there is no further flow of water, no further flow of infection. Now, so this was all about, we have talked about wood, we have talked about secondary growth in xylem. I just want you all that after learning the secondary growth and after learning about this wood, you just try to observe around yourself and try to analyze the wood. If it's possible, try to analyze the wood which you see around you. Even in your furniture also, if you try to analyze them, you can make out some difference, and try to find out that what type of wood is there, whether some rings are there, growth rings are there, whether like if you some trees there around you, and if it is cut, you can analyze them also. So, if possible, you try to analyze the wood from your side and try to find out that what type of wood it is and what might be the difference in its characteristic features. So, thank you for today.